Biochemistry.
Yay!
Alright, well, we're going to go over the different levels of structure.
Oh, I love chemistry. Yay!
Let's do a very, very fast and quick protein review because you, my friend, can go and look at the other videos for all this. But just from the start, proteins are made of a chain of a very specific sequence of amino acids. Amino acids, remember, have a central alpha carbon that is always going to be bonded to a hydrogen, an alpha carboxyl group, and an alpha amine group. Okay? The other group, because remember, carbon likes 4 single bonds, or if it has double, or who cares about that, but our amino acids have central carbons with single bonded groups, and the last one is going to be the one that changes, and we're going to call that the variable R group. That's what's going to give each amino acid its own little function, its own little place in the world, whatever, of proteins. So those are our amino acids. They're going to be bonded together into a polymer, poly meaning many, chain of polymer. The polymer is going to be called a polypeptide. Once it's functional, then we call it a protein. Okay? Now we have 4 levels of protein structure. We have the linear sequence. Sequence is going to equal the function. So structure equals function. The initial structure being in the sequence is going to determine exactly how this protein folds and exactly how this protein is going to act once it's functional. The amino acids are going to be bonded together with amide bonds. We're going to call these specifically peptide bonds. So when we bond two amino acids together, we're going to call that a peptide. Okay, a dipeptide specifically, and then when you get so many in the chain, then we call it a polypeptide. Polypeptide, it is a directional molecule, it's an informational molecule, or an informational polymer of one of the biomacro molecules. So NC-alpha C, then it's peptide bonded, the alpha carboxyl group is bonded to the next amino group, the nitrogen, NC-alpha C, that's our polypeptide. We're going to call this linear sequence of the NC-alpha C is the backbone of the protein. And then we have the functional R groups that are going to determine basically how it folds. Its native conformation will be the eventual folding pattern of each individual protein. This individual peptide bond, the actual peptide bond does not permit rotation, so it's because, remember, the resonance structures. But the other bond, the C-alpha C carboxyl group bond, and then the C-alpha N bond, both of those do permit rotation. The C-alpha C bond is going to be what we call the psi bond, and then the C-alpha N bond is going to be the phi bond. These 2 bonds in any given amino acid are going to be where the rotation, the movement can occur. And so the conformation will ultimately be determined by the phi and psi bond. Now there are different kinds of bonds in the protein folding, and as we go over the different structures, the secondary structure and the tertiary structure, we'll hit on exactly what's going on. But let's just look at a diagram where we can see the different kinds. We have ionic bonds, these are non-covalent. The hydrogen bonds, these are going to come into play everywhere, not only within the protein, but with the protein and the aqueous environment that it's interfaced. And we have some van der Waals attractions as well. Another kind of bond that we'll see is a disulfide bond, which is covalent. This is going to be between 2 cysteine residues. We also have hydrophobic attractions, and this is going to be between the hydrophobic molecules, and this is going to tend to be in the core of a protein, of a globular protein. Now, in any given protein, the polar side chains are going to be interfacing with the water, the aqueous environment, so you're going to find those on the outside of a globular protein. If we have a transmembrane protein, that's going to be a little different, because where would the polar amino acids be with the embedded protein in the membrane? We've got to think, what is it embedded in? It's embedded in the fatty acids. So it would make sense that the hydrophobic molecules are going to be interfacing with the lipid bilayer, the fatty acids that are hydrophobic. So in this case, in a transmembrane protein, it is going to be on the outside of a globular protein. Now, where the protein's sticking out into the interstitial fluid, or the extracellular fluid, or the intracellular fluid. Now, on the tips of the transmembrane protein, we're going to have some polar amino acids. So it just makes sense Remember, hydrophobic likes hydrophobic and hydrophilic likes hydrophilic. So we have a protein. Think of a pearl necklace, okay? I take my pearl necklace off and I throw it, or maybe Mardi Gras beads. I don't know, whatever you want to think of. And if you throw it, it's going to land in a certain little jumbled up conformation. Now, if you do it again, is it going to be in the exact same conformation? Not if you're Mardi Gras beads. But if you're a protein, yes. Every single time it is going to fold in the exact same conformation as it did before. Why? Because we have a linear sequence of amino acids, and those residues on the amino acids are going to make sure that it folds in the exact same way. It's very, very, very important, the linear sequence of amino acids. You take one of the linear sequence and you mutate it, you switch it with something else, you can end up with sickle cell disease, which is very, it can be deadly. It's a horrible disease. So one amino acid off of the chain and you've got big problems. So that's why the structure, the linear sequence, which is the primary structure, is extremely important. Now, in this native conformation, this is where the free energy is going to be the most energetically. Now, chaperones are going to be proteins that are going to, they're favorable proteins. They kind of look like waste baskets in a way. You're going to have a polypeptide chain. Remember, the ribosomes is where it's made from, and it dumps off into the RER. Then we're going to have these chaperones, which are proteins that are going to assist in the proper folding. Now, would a protein fold on its own? Sure. But a chaperone is going to speed it up and make sure that the folding is done in the most energetically favorable pathway.
Secondary structures make my whole day.
Moving on to Alpha Helix. Woo! Alright, so we have two regular folding patterns in secondary structure. Now we're moving on from the linear sequence, which was just the exact sequence of amino acids. That's linear sequence, that's primary structure. Secondary structure, now we're going to have some localized bonding, hydrogen bonding, if you remember, from Unit 1 of my biochemistry series, and if not, go back and watch it. But hydrogen bonding is going to come into play with the localized interactions in secondary structure. Secondary structure, the first one is alpha helix, the second one is the beta pleated sheet. In essence, secondary structure is simply regular folding patterns that you're going to find in any. The secondary structure does not involve the amino acid residues. Those don't come into play until tertiary structure, which is the next level. Talk about the alpha helix. There's two different kind of regular folding patterns that we'll find with these localized hydrogen bonds. And the first one being the alpha helix. It is, if you know the structure of DNA, it's kind of like that. It's a coil. It's not a double helix, it's just a single helix. But the amino acid residues, every fourth amino acid is going to hydrogen bond. The NH bond of one amino acid is going to hydrogen bond with a carbonyl 4 amino acids down the chain. And so this is going to cause a coil. Okay, this coil, there's going to be a turn every 3.6 amino acids. So this is our alpha helix, okay? So every fourth amino acid is going to bond the NH to the carbonyl, okay, hydrogen bond. Remember, these are very weak bonds, but it's there nonetheless. And so this causes a turn every 3.6 amino acid. Now again, this is only involving the polypeptide backbone. This does not involve the residue. We're going to find a lot of alpha helices in proteins, especially ones that are involved in cell membranes, like transport proteins, receptors. Some of these transmembrane proteins are going to be characterized by the amount of alpha helices that are going across the membrane. Alright, so the beta-plated sheets, this is another secondary structure. And remember that a protein is directional. So we start with an N-terminus, and then we end with the C-terminus. So NC-alpha C, NC-alpha C, etc. So it is directional. This is an informational molecule. It's not, you know, like a big chain of carbohydrates that tells you nothing. There's information in this protein molecule. Beta-plated sheets are going to be more structural. You're going to find these in the core of proteins. Alright, two different kinds of beta-plated sheets, two different main kinds. We have parallel, so this would make sense that we're forming from neighbors that are going in N-terminus to C, N-terminus to C. So they're going the same direction, and that's going to be a parallel sheet, which is not going to be as stable as a beta sheet that is basically folding upon itself and then hydrogen bonding between, and that's called an anti-parallel, because if you go N to C and then you're going N to C, N to C, N to C, so it's anti-parallel. This is actually the more stable of the two kinds of beta pleated sheets. The parallel sheets are going to be less common, of course. They're going to not be found in great amounts by each other because they're less stable. So anti-parallel is the stable form of the beta pleated sheet, and the parallel ones are not. We have what is called the beta strand, which I should have mentioned before, which is about 3 to 10 amino acids long. And so these beta strands are what is forming. If you kind of think of an accordion, how it kind of folds like that, but it's different strands, beta strands, bonded together through hydrogen bonding. Now let's look at some super secondary structures. Now the hydrogen bonding of the beta strands are not always perfect. They can have local disruptions, and we're going to call these beta bulges. A beta bulge is usually going to be by inserting a residue with a helical dihedral angle into, one of the hydrogen bonded beta strands. A beta bulge can even cause the beta sheet to fold over and cross itself. The most basic function of a bulge is going to be to accommodate an extra residue that possibly has been added due to a mutation, but it still maintains the bonding pattern and thus the overall architecture of the actual protein. Sometimes these bulges are going to help form the structure of an active site, like in an enzyme. So the beta bulges are going to be contributing to the ultimate protein structure. We have a few common structural motifs. A motif is not going to be functional, but it's going to be a structure that you're going to find to be common. And a very simple structural motif would be that involves beta sheets is going to be the beta hairpin. We have 2 anti-parallel strands linked by a short loop of up to 5 residues. Frequently, this is going to be a glycine or a proline. These are going to cause the loops. Another motif is going to be the Greek key motif. This is 4 adjacent anti-parallel strands, and of course they're linking loops. So we have 3 anti-parallel strands connected by hairpins. Remember, the hairpins are going to be the glycine or the proline typically. And the 4th is going to be adjacent to the first and linked to the 3rd by a longer loop. This structure is going to form during the protein folding. And the reason why they call it a Greek key is because it's like the ornamental artwork found in Greek vases. Okay, we have the beta alpha beta motif. It's due to the chirality of the amino acid residues. So all the strands are going to have a right-handed twist, and the linking loop between the 2 parallel strands almost always is going to have a right-handed crossover chirality, which is going to be favored by the inherent twist of the sheet. We have the beta meander motif. This is a simple supersecondary protein structure. We have 2 or more consecutive anti-parallel beta strands linked together by hairpin loops. The beta meanders can be found in several structural architectures such as the beta barrels or beta propellers. And again, these are all just super secondary structures. We have the psi loop motif, and this is 2 anti-parallel strands with 1 strand in between, connected to both by hydrogen bonds.
I love tertiary structures.
Now it's time to talk about tertiary structure. Yay! Okay. So tertiary structure is going to be the more 3D folding. We're going to start to come into play with the amino acid residue. So this is where the sequence of amino acids is really going to come into play on how this protein is going to fold in its native conformation that is energetically favorable. So it's going to have the lowest free energy with a functional protein. Now, tertiary is the first level of protein structure that can be functional, but they're not all functional because some of them need to go to the quaternary level if they're going to be in complexes. But a tertiary is going to be a 3D structure of a protein that we're turning a polypeptide. It's growing up. It's going to grow up and it's going to get a job, and its job is going to be its function. It's going to be awesome. So whatever it is, whether it's an enzyme, receptor, a hormone, see if I can remember them, cell signaling, structural we have, transport across the membrane, globular protein, roughly spherical protein. If it's in an aqueous environment, what is going to drive the folding process is the hydrophobics. Why? Because they're so scared of water. They're so scared. So they want to get away from water. It's like all of these people running from Godzilla, with water being Godzilla, and they're running together, so they stay together, and so they're going to run into the core of the proteins. So the hydrophobics are going to drive the ultimate 3D folding and of a globular protein in an aqueous environment, which the body is an aqueous environment, so it's pretty much what it is, but we will have an exception to this with a case of a transmembrane protein in a minute. But the hydrophobics are going to make up the core. Sometimes they're going to make up part of the active site, okay, and it depends, an active site of an enzyme. So you're going to have a glob of kind of like a micelle. If you kind of remember the structure of a micelle, remember the micelle is not amphipathic molecules with the fatty acid chains on the inside and the hydrophilic heads on the outside that are interfacing with water. It's kind of like that. That's kind of what a protein is. It's kind of a glob of hydrophobics on the inside with the hydrophilics on the residues facing the water. It's kind of like that. So all of your polar compounds are going to be on the outside or in the active site, and then the hydrophobics are going to be in the core away from the water. So that's going to be basically how a globular protein is going to situate itself. The exception to that is going to be a transmembrane protein. Think about, and this is what I was talking about a little bit ago, think about what do the hydrophobics want to be around? Well, they have hydrophobic interactions, meaning that if you're all scared of Godzilla, then you have something in common. You want to be around each other and vent about how scary that big dinosaur thing. What was Godzilla anyway? It's like a big lizard? I don't know. But you know, you have something in common with each other, and you're afraid of the same thing, so you want to be around each other. So the hydrophobics are going to now, because they're also fatty acid chains inside of a lipid bilayer, these fatty acid chains in the middle of this lipid bilayer, so this transmembrane protein, what is interfacing with the fatty acids is hydrophobics. So it's kind of like this protein has been turned inside out. Now all of a sudden the hydrophobics are on the outside. If these were hydrophilics, would it stay embedded in a hydrophobic inside of a membrane? Heck no, it's going to pop out, and it's either going to go to the ICF or the ECF. Okay, so in order to anchor a transmembrane protein into a membrane, we have to have the hydrophobics on the outside this time. Say you're a channel protein. Okay, this is one of the transport proteins. If you're a channel protein, then what you have is a channel that goes down the center of your protein. Now in this channel, we're going to have the hydrophilic or the polar molecules making up this aqueous environment of the channel. And that's, so say certain ions can pass through the lipid bilayer. Okay? So a channel protein that's embedded in the membrane is going to be an exception to the rule of having a hydrophobic core and a hydrophilic surface of a globular protein. I hope that makes sense and I didn't bother you. Okay, so in essence, the tertiary structure overall is not going to be as stable as, say, the secondary structure, and of course the primary structure, which is linear sequence. Linear sequence is all covalently bonded. That's going to be really strong. Secondary, because it's localized hydrogen bonding, that's going to be more stable than the 3D folding of the tertiary structure. But there are a lot of different kinds of bonds at play here. So we have the hydrogen bonds, not just the localized, but as the hydrophobic molecules come together, it might put two molecules that are, you know, partially negative, partially positive, close proximity, and they'll form another hydrogen bond. Even though hydrogen bonds are weak, power in numbers, if there's a lot of them, then it's going to help the strength of tertiary structure. Then we have the ionic bonds. We have a negative acid, a positive base residue coming into close proximity, and they will be attracted. We also have the disulfide bridges made from the cysteines, the thiol, the SH groups of each cysteine residue. If they come into contact, we'll make a sulfur-sulfur bond. That is a covalent bond, so that's relatively strong. The more disulfide bonds, the better. Obviously, the more strong your tertiary structure will be. And then we also have metallic bonds. So you might find a magnesium stuck in between part of the polypeptide chain bonded.
This is Brenda, the not-so-good witch, signing off for today. See you next time on Dr. Bond's Science Theater.